Hello and welcome to Biomed Vibes. Today we are going to talk about the extracellular matrix. If we begin at the plasma membrane and moving outward, we can see an organized network of secreted molecules that provides a scaffolding to the cells and tissues it surrounds. The extracellular matrix is not all about scaffolding. It also provides physical, biochemical, and mechanical signals that play an important role in the cellular activities and shape. One of the well-known extracellular matrices is known as basal lamina or basement membrane. Basal lamina or basement membrane. The basal lamina is found almost most parts of the body, from, let's say, outside. The fat cells, muscle cells, and also in the inner endothelial blood vessels, and also it underlies in the digestive and respiratory tracts. As you can see, we can see the basal lamina in almost most parts of the body. We are also going to talk about the components of the extracellular matrix. We are going to talk about four major types of components which comprise the extracellular matrix. We are going to talk about collagen, proteoglycan, fibronectin, and also laminin. Okay, I'm going to clean the board and talk a little bit in details about each one of these. Okay, let's talk about collagen. Collagen is a family of fibrous glycoproteins. Glycoprotein actually is a protein which contains carbohydrate chains in its structure. The collagen gives the cells tensile strength and also it is produced by mainly by fibroblasts and also epithelial and muscle cells. But let's talk about the structure of the collagen. The collagen consists of 3 polypeptide chains known as alpha chains. 3 polypeptide chains, fibrous, known as alpha chain. These chains are mainly consist of proline and some lysine residues which are hydroxylated the following synthesis of the polypeptide. Let's say the synthesis of the polypeptide. The proline, mainly proline, is involved in the synthesis of the polypeptide. It has to be mentioned that failure to hydroxylated chains may lead to the scurvy disease. Scurvy disease is a disease which is a consequence of the lack of ascorbic acid or the vitamin C. What vitamin C does to this to the collagen is that the vitamin C acts as a coenzyme with the enzymes that are needed for the production of the polypeptide chains, of alpha chains, let's say of alpha chains. The lack of vitamin C may lead to the scurvy disease. I'm not going to talk about this disease, but it results in a lack of ascorbic acid. Okay, let's talk about the proteoglycan. The proteoglycan consists of a core protein as well as a lot of glycosaminoglycans which are covalently attached to the core protein. Glycosaminoglycans consist of repeating polysaccharides, and they consist carboxyl and sulfate groups, which these two are highly acidic. It turns out that the negatively charged glycosaminoglycans can lead to the absorption of a lot of cations. For example, water molecules can bind to these glycosaminoglycans and form the hydrated gel, which make the proteoglycan with collagen together to give cartilage and strength, and also make them to be resistant to the deformation. Also, the proteoglycan involves in cell signalings such as FGF, VEGF. FGF stands for fibroblast growth factor, and VEGF stands for vascular endothelial growth factor. The proteoglycan, as I told you earlier, consists of a core protein as well as a lot of glycosaminoglycans. So, core protein as well as a lot of glycosaminoglycans. As I said before, residues and these. As I said before, because of the negatively charged glycosaminoglycans, they turn out to attach to a lot of cations such as water molecules and form a hydrated gel that give a strength to the extracellular matrix. Now we are going to talk about the laminin and fibronectin. Okay, let's talk about fibronectin. The fibronectin consists of similar but non-identical polypeptides that are joined by a disulfide bond together. Let's draw what this sentence states. Let's draw polypeptide chains. There should be similar but non-identical here. Here we have our C-terminal and N-terminal of our polypeptide. These bind by a disulfide bond, actually a disulfide pair. A pair of disulfide bond. Here and here, for example. Okay, it's not clear, but I try to. Each polypeptide is composed of linear series of distinct modules, which are organized into several functional units. For example, this unit may call, let's say, fibrin binding site. This may be collagen, for example, collagen binding domain. There are a lot of different domains and binding sites here. For example, heparin here, heparin binding site. There are a lot of binding sites in the units of the fibronectin, which are very crucial for the extracellular activities. Also, the fibronectin leads to the growth of the cells, to the differentiation of the cells, as well as migration. Now let's talk about laminin. Laminin consists of 3 different polypeptide chains. It has 3 short arms and one long arm, as you can see here. Let's see here. Let's consider it is the long arm and 3 short arms known as alpha, beta, and gamma. The laminin influences the cell potential for growth, differentiation, and migration. As you can see, it is all the same as the fibronectin function. These are the same. Also, the laminin together with type 4 collagen form separate but interconnected network. Together, laminin with type 4 collagen, they form separate but interconnected network that gives cartilage and strength to the extracellular matrix. They bind together via a protein known as entactin. For example, here let's see, we have our laminin, and here we have our collagen. It's more complex than that, but the idea is that they form an interconnected network via a protein known as entactin.